For thousands of years, humans have relied on selective breeding, waiting generations for the right traits to appear in crops or livestock. But CRISPR changes that entirely. It allows us to directly edit the code of life in real time. This technology is remarkably accessible. You can currently order a basic bacterial gene editing kit online for roughly the price of a mid-range smartphone. Jennifer Doudna and Emmanuelle Charpentier were awarded the 2020 Nobel Prize in Chemistry for proving that this process could be harnessed to reprogram the genomes of living cells. This wasn't a human invention. Bacteria have used this system for eons as a molecular vaccination card, storing snippets of viral DNA to recognize and destroy future infections. By hijacking this ancient microbial defense, we have transitioned from being the observers of biology to its active authors. This diagram illustrates the two-part CRISPR-Cas9 mechanism. A guide RNA searches for a specific genetic sequence, and when it finds a match, a protein called Cas9 severs the DNA at that exact location. When the cell rushes to repair this break, it often does so with a small error. This effectively breaks the gene, creating a knockout that disables its function. If researchers introduce a new piece of genetic code during the repair process, the cell incorporates that new sequence into its DNA. This knock-in allows scientists to add entirely new traits. This programmable accuracy has turned gene editing into a surgical process. We can now target a single base pair among the 3 billion that make up the human genome. This precision is already being used to treat monogenic disorders, diseases caused by a single identifiable mutation. Clinical trials are currently targeting the specific genetic switch that causes sickle cell disease. These treatments use somatic editing. The genetic change stays localized to the patient's own tissues, such as their blood or immune cells. Because these edits don't affect the reproductive system, they cannot be passed to the next generation. In germline editing, the tool is applied to an embryo. The edit is copied into every cell of the developing adult, meaning the change is inherited by all future descendants. Somatic editing cures the individual, but germline editing allows us to actively design the future of the human species. In 2018, scientist He Jiankui bypassed international ethical norms and announced the birth of the world's first CRISPR-edited human babies. He targeted the CCR5 gene in human embryos, attempting to grant the children a permanent, heritable resistance to HIV infection. The backlash from the scientific community was immediate. The experiment demonstrated that our technical ability to use CRISPR is currently moving much faster than our ability to regulate it. While resistance to a virus involves a single gene, complex traits like memory or intelligence are polygenic, the result of thousands of genes interacting in ways we don't yet fully understand. And yet, despite this complexity, the successful edit of a human embryo has moved the conversation toward elective enhancement. These enhancements will likely be expensive, raising the prospect of extreme economic disparity. In this scenario, a family's bank account could determine the cognitive or physical traits of their children. If elective enhancements remain unregulated, we risk turning temporary wealth inequality into a permanent genetic divide. While human enhancement remains controversial, the animal kingdom has become a primary testing ground for CRISPR's most ambitious applications, including de-extinction. Geneticist George Church and the company Colossal are currently working to resurrect a functional version of the woolly mammoth. Asian elephants and extinct mammoths share 99.6% of their DNA. The project focuses on the razor-thin 0.4% that separates them. Using CRISPR, scientists knock in mammoth genes for shaggy hair and subcutaneous fat, creating a cold-resistant hybrid. This isn't for a zoo. The goal is to return these hybrids to the wild to serve a specific planetary purpose. The Arctic permafrost currently traps billions of tons of carbon. As temperatures rise, this landscape is thawing, threatening to release those gases into the atmosphere. Reintroducing heavy, cold-adapted herbivores could help prevent this. By packing down snow and restoring grasslands, these hybrids keep soil colder and absorb more carbon. This is a radical escalation of the technology. We are moving from microscopic DNA edits to the active restructuring of the Earth's climate systems. A defense system borrowed from bacteria is now being used to rewrite human health and manage planetary ecology. Our technical capacity to edit the biosphere has arrived. The remaining variable is whether we can develop the governance to match that power,